Welcome. Today, I will talk about the expression and function of L-type voltage-gated calcium channels in neurons and their role in Parkinson's disease. L-type or Cav1 channels are members of high-voltage activated calcium channels family. They activate at strong membrane depolarizations and inactivate slowly, producing a sustained calcium influx, hence the L for long-lasting. The core of the L-type channels is the alpha-1 subunit, which forms the ion-conducting pore and contains the voltage sensor. Channels trafficking and kinetics are fine-tuned by its beta and alpha-2 delta auxiliary subunits. L-type calcium channels are encoded by 4 genes that produce Cav1.1, Cav1.2, Cav1.3, and Cav1.4 subunits. Cav1.2 and Cav1.3 are the main L-type calcium channels in the brain, while Cav1.4 is restricted to the retina and Cav1.1 is expressed in non-neuronal cells. L-type channels are located on neuronal soma and along dendritic processes. In neurons, L-type channels translate membrane depolarization to long-term changes by triggering calcium-dependent signaling cascades. For example, L-type voltage-gated calcium channels shape neuronal firing by modulating membrane depolarization. Moreover, the calcium influx through these channels tunes synaptic plasticity, a process that underlies long-term potentiation and depression involved in learning. At presynaptic terminals, L-type calcium currents trigger neurotransmitter release, and when they reach the soma and nucleus, they activate kinases and transcription factors to shape gene expression. Dysfunction of L-type calcium channels has been observed in several diseases. Moreover, preclinical studies have demonstrated that isradipine, an L-type calcium channel blocker, exerts neuroprotective effects in Parkinson's disease models. Parkinson's disease is a progressive neurodegenerative disorder characterized by the loss of dopaminergic neurons in the substantia nigra, leading to motor symptoms like tremor, rigidity, and bradykinesia. In Parkinson's disease, activation of Cav1.3 channels in pacemaking dopamine neurons leads to excessive calcium influx that could cause mitochondrial dysfunction, oxidative stress, and neuronal degeneration. Despite strong neuroprotection in animal models, the Study Parkinson's Disease 3 clinical trial of isradipine failed to slow motor progression at the doses tested. That's all for today. Thank you for watching, and don't forget to subscribe to learn more.